Okay, this video is called Biology Ruler. And the whole point of it is just to give you a sense of the different size of things related to keeping a cell alive, keeping you healthy. I think you're going to find this really useful. If you care about physiology and understanding what's going on at a cellular level, this is useful stuff. So here's a molecule of glucose. This is in a Hayworth projection, like a ring projection. I remember that because Rita Hayworth was a movie star. The areola of her breast is round. So that's how I remember a Hayworth projection, okay? The point is it's 1 micron, okay? We're going to go through some real common stuff and talk about the size. So here's glucose in a Fisher projection. I remember Fisher projection because you take a fishing line, and so it's a straight line. So it just means the ring is opened up. So you take a fishing line, that's a Fisher projection. That's how you can remember it. And I just want to talk about the difference between glucose, which is your carbohydrate. Your polymer of glucose is a starch. And palmitate, which is your standard most common fatty acid in the human body. It's a C16, 16 carbon fatty acid with no double bonds. So it's got a lot more carbons than glucose. Glucose only has a 6 carbons. This has 16. So this is twice as big, though, in biology terms, 2 microns, okay? And that's a standard, fatty acid that could be in your plasma membranes. Now here is the glycocalyx. Glyco means sugar, calyx means coating, and because you got repeating disaccharide polymers as part of your glycocalyx. All it means is it's the coating that sticks up like trees on a hill. So this would be like the inside of a capillary, and these are the glycocalyx. They stick up like little trees or little plants coming up. Okay, all this is going to make sense in a moment, but the key to know is this is about 600 microns, and I'll show you how that relates in a sec. This is you're going to be surprised how useful this is. There's a reason why I'm showing this to you. Okay, so here is the simple plasma membrane, which is just a phospholipid bilayer. There's a phosphate head, and those are lipid tails on them. Okay, they look like little sperm with 2 tails. That's only 6 nanometers, okay? Then there's going to be adhesion molecules. They're not drawn in here. They're going to stick up about 60 nanometers, so 60. And then the core protein and the glycocalyx, that's going to stick up about 600 nanometers. So it's actually kind of like a devil number, 666. 600 for the core protein, going to be 60 for adhesion molecules, not drawn in this picture, and then 6 nanometers. All of this in nanometers for the plasma membrane, okay? Glyco in the sense that heparan sulfate is repeating disaccharide. At the end of it is a sialic acid. That's the immune system identity card, okay? And the reason it's slippery is because the sulfates give it a negative charge. Water has a dipole, positively charged hydrogens, negatively charged, partial charges on the oxygen. And so the positive charge of the hydrogen line up with the negative charge of the glycocalyx, which is really also a zeta potential, you could call it. And that's why you say slippery when wet. They make it more slippery so the blood cells can travel through the capillary more quickly along the endothelial glycocalyx. Okay. Trust me, this is all going to come together and make a lot of sense here in just a moment. So here's a drawing from a research article about it. So here's the glycocalyx right there. This sticks up about 600 nanometers. Okay, that's worth knowing. And then beneath that are these smaller adhesion molecules. These are only sticking up, like ICAM is intracellular adhesion molecule. VCAM would be vascular cell adhesion molecule. Those are only about 60 nanometers in height, so about 1/10 the height of the glycocalyx. When the glycocalyx is destroyed or shed away for whatever the reason, these adhesion molecules will now be exposed and they will bind to white blood cells. Okay, they can also bind to red cells, some of them, and cause a clot. Okay, so you don't want them exposed unless there's a major problem. So here's thrombus, a clot forming around these adhesion molecules. Okay. Now here's an example when you eat a high-fat meal, what happens is the fat, like sat fat for example, will activate your neutrophils. They'll degranulate, release MPO, myeloperoxidase. It's very positively charged, that means cationic as well. The positive charges of the myeloperoxidase, MPO, will bind to the negative charges of the glycocalyx. It's zeta potential is the word for negative charge, and they will cause it to collapse down. When the glycocalyx collapses down, these green things here are adhesion molecules. They will become exposed. And when the adhesion molecule is exposed, now the neutrophil, NP is for neutrophil, can bind to it. So you don't want your white blood cells adhering to your endothelium. So that's another reason why you don't want to eat, meals high in saturated fat. Okay, so anyways, that's how that works. And the 666 mnemonic means that the glycocalyx is 600 nanometers in height. These Adhesion molecules are 60 nanometers in height, and the plasma membrane is about 6 nanometers in height. So it's easy to remember, 666. So here's these rules. I'm going to show you some tricks for memorizing all this stuff. So whenever you're reading about these things and you hear sizes, you'll be able to make sense of everything very quickly. Glucose, 1 nanometer in size. Palmitate, 2 nanometers in size. So when you're talking about a single molecule, you're talking nanometer sizes. When you talk about a plasma membrane, here's the trick. 600 nanometer glycocalyx sticking above it like a tree sticking up from the endothelial cells. The adhesion proteins are just 60 nanometers, so 666, and then the plasma membrane itself is just 6 nanometers. So this stuff again is in nanometers. All right, now when you're talking about living things that are relatively independent, a bacteria is 1 micron, and it makes sense that mitochondria are thought to be derived from bacteria, so they're also 1 micron. Okay. A capillary diameter is about 5 microns. A red blood cell is a really small cell. It's just a little bag full of hemoglobin. That's 7 microns in diameter. Now, a neutrophil, which is the most common white blood cell in the blood, is about 14 microns. Okay, you just double the RBC to get 14. And now you're going to get into the rule of 4s. The rule of 4s means that a neutrophil is 14 microns. Add 4, you get a monocyte. 18 microns. Add 4, you get a macrophage. 22 microns. Okay, the relevance is neutrophil is most common, and it's much, much bigger, twice as big as the red blood cells. That's why it's got that elongated, segmented nucleus that it can get through the capillary, because it has to really elongate itself like a torpedo. All right. Now, a monocyte is really like a precursor to a macrophage. Macro means big eater, but you really can't be that big inside the blood vessel. You won't be able to get through those capillaries. They're only about 5 microns in diameter. When it gets extracellular, the monocyte enlarges and transforms into a macrophage, about 22 microns. Okay. And then a real important cell in the body is a liver cell, and that's about 26 microns in diameter. Okay. So these are numbers worth knowing. If you're going to do a lot of molecular biology, having these numbers in your mind, everything will make sense to you very quickly. Endothelial cell, this one's not that important to know, but just know that it has a length about 50 microns. It's pretty big, okay, and it's shaped like a spindle along the long axis. Is long axis in the direction of flow. You don't really need to know that so much. Just for a little perspective on sizes too, a human hair is about 50 microns thick, okay. So these are basic numbers. I find it helpful to know this stuff because I spent a lot of time reading about the physiology of these areas. So I just in my mind's eye have a sense of all the sizes, and it helps me to get through the papers and stuff. And it'll make concepts make a lot more sense if you know that. You don't need to know it, but I think it's nice to know it. So when you're hypertensive, the hypertension high velocity blood flow hits the median divider between the branch point, and then it bounces off. Excessive turbulent flow, chaotic flow, and retrograde eddy currents. They will confuse the endothelial lining cells, and they will shed their glycocalyx and expose these adhesion molecules, and you can form clots right there. In addition, when you expose these adhesion molecules, circulating cancer cells in the blood can adhere to them and then work their way into the subintimal space and produce metastases. So what I'm saying is hypertension damages the endothelial glycocalyx in a way that not only promotes clots and subsequent atherosclerosis, it also increases the risk of metastatic cancer. So that's, the same risk factors for hypertension and diabetes and atherosclerosis are risk factors for cancer. And I'm explaining to you from a blood flow point of view, this is one of the reasons why. Okay, here's just showing you some blood cells like I was talking about a moment ago, that the red cell is about 7 microns. You can see a red cell right here, a little bag of blood. Neutrophil is about twice as big. It's got that, polymorpho nuclear leukocyte. Leukocyte means white cell. Polymorpho nuclear means its nucleus is this unusual shape, segmented nucleus. And that enables it to stretch out like a torpedo and get through the capillaries despite its big size. 14 micron size, getting through a 5 micron capillary. And then the monocytes are even bigger. Okay, they're like about 18 microns. Once they get outside, they can turn into a macrophage and be about 22 microns. So anyways, I hope you found that helpful, the concept of the relative sizes. And the rule of 6 is for the glycocalyx, 666, and the rule of 4s for the blood cells.